Hi, I'm Sanjida from the NCBI at the National Library of Medicine at the United States National Institutes of Health. Today, I'm going to tell you about our brand new tool, Comparative Genome Viewer, or CGV. This tool allows you to view assembly alignments generated in-house at NCBI. Here, I filled out a selection menu to select two different sheep assemblies to compare, or Rambouillet V10 and ARS UI Ram V2.0. Now, let's look at the viewer. You'll see the two assemblies with their chromosomes laid out horizontally. Colored lines or blocks are assembly alignments, that is, regions of sequence match between the two assemblies. I'll click on chromosome 11 to zoom in further, and I can see that there's a region of similarity between chromosome 11 on this assembly and chromosome 18 on the other. This may be a genome rearrangement, a translocation. If I click on chromosome 18 and zoom in even further, I'll be able to view and compare gene annotation between these two assemblies. I can also click on an alignment block to view a box with more information about it. We're still a brand new product, so please click on this feedback button if you'd like to get in touch with ideas about how to make us better. CGV is part of the NIH Comparative Genomics Resource, a project to establish an ecosystem to facilitate reliable comparative genomics analyses for all eukaryotic organisms. Thank you for your interest.